In this section of the lecture, we're going to be looking at regulation by negative control, and we're going to learn more about the process of repression and induction. So this is the first example of negative control that is going to be repression. And repression, it is a mechanism in which transcription is repressed in the presence of a co-repressor. One operand that exemplifies this mechanism is the arginine operand. Now, what we have over here is an illustration of the arginine operand, containing in yellow the arginine promoter, a region of the arginine operator, and three genes that are involved in arginine biosynthesis, arginine G, B, and H. In the absence of the amino acid arginine, the RNA polymerase is able to bind to the promoter and initiate transcription to make the enzymes RGC, RGB, and RGH. The repressor, it's not going to bind. But once the arginine has been made, and now there is presence of arginine, arginine behaves as a co-repressor. So the co-repressor is going to bind the repressor that's going to have a conformational change, and that conformational change is going to allow the co-repressor repressor dimer to bind to the arginine operator. The arginine operator now, the binding of that co-repressor repressor dimer inhibits the RNA polymerase, and therefore transcription is blocked. So we have repressed the expression of the arginine operand. So for example, in this graph, we're looking at the relative amount of three different factors: number of cells in red, the total amount of protein in black, and the amount of the arginine biosynthesis enzymes in blue. And at the bottom is going to be time. So at the beginning, when arginine is not present, you can see that the cells are growing and the number of cells is increasing. The total amount of protein, in those cells also increases with increasing cell numbers. But notice that the amount of the arginine enzymes goes up and continues going up until the arginine is added. At that point, there is no more transcription of the arginine operand, and the amount of enzymes never increases as long as arginine is present. So for repression to work, we need to have a repressor protein that is going to bind to an operator, and binding of that repressor protein to the operator depends on the presence of the co-repressor, in this case, arginine. You're going to see that this repression mechanism is going to be used by a lot of operands that are involved in the biosynthesis of amino acids and other substances. Another example of negative control is induction, and that process of gene regulation is exemplified by the beta-galactosidase operand, or the LAG operand. So the LAG operand also contains a promoter. It contains an operator region next to the promoter, and three structural genes: the LAG-Z gene, the LAG-Y gene, and the LAG-A gene. The LAG-Z encodes for the enzyme beta-galactosidase, the LAG-Y is the lactose permease, and the acetyltransferase is the LAG-A. Now, we also have another gene that is required in this, which is the repressor. And the repressor protein is encoded by the gene LAG-I. And the repressor, it has its own promoter as well as gene expression that is independent of the LAG operand. So this operand is in a state of block transcription, because the repressor can bind to the operator, and binding of the repressor to operator inhibits transcription. So now we're going to see that in the presence of lactose, lactose now behaves as an inducer, and lactose then binds to the repressor, induces a conformational change, and that conformational change allows the repressor to fall off. Now, the RNA polymerase is able to initiate transcription, resulting in the expression of the LAGZ gene. So now, I want to illustrate this over here as an example for induction. Like in the previous graph that we saw, we have the relative increase in the y-axis and time in the x-axis. And we are still going to be looking at the number of cells, the total amount of protein, and in blue, we're going to be looking at the amount of beta-galactosidase, which is the product of the LAGZ gene. So as you see, with time of growth, the number of cells increases, and the amount of total protein also increases. But notice that the amount of beta-galactosidase is not present until lactose is added. And at that point, the amount of beta-galactosidase goes up. So just to remind you, the enzyme beta-galactosidase is able to hydrolyze lactose into two sugars, glucose and galactose. And that is illustrated here by this picture where you can have the lactose disaccharide, which is going to be hydrolyzed into galactose and glucose by the enzyme beta-galactosidase. And this enzyme is only found intracellularly in microorganisms. The interesting thing about the lactose operon and many other operons is that it displays what is called a dioxys growth phase, meaning that in the presence of glucose and lactose, dioxys growth is going to have a double growth. That it's going to say that you're going to have a growth curve with the utilization of glucose and then a different growth curve with the utilization of lactose. And that provides, as you see here in this image, two different growth phases. In red, we have the growth of lactose, and you can see the amount of and you can see the number cell density going up really quickly with a very steep slope. When glucose is exhausted, the growth becomes now lag and it doesn't change anymore. And then the cells begin to grow now with utilizing lactose. The utilization of lactose follows the amount of beta-galactosidase present. So at the beginning of culture, you can notice that when glucose is being utilized, there is no beta-galactosidase present in the culture. But after this lag phase, when glucose has been exhausted, the enzyme beta-galactosidase levels increase, and that is illustrated also by the amount of growth of lactose. So catabolite repression, it is an effect in which glucose becomes the preferential sugar that is going to provide carbon and energy for cells, inhibiting the use of the other sugar. And we're going to see that lactose and many other operants are subject to catabolite repression. So how is this happening? Remember that we learned about the glucose PTS system, which is involved in the transport of glucose into the cell. Well, that PTS system has a capacity to control the synthesis of beta-galactosidase mediated by the amount of cyclic AMP present in the cell. So when glucose levels are high, we're going to have a low amount of cyclic AMP. Whereas when glucose levels are low, we're going to have high amounts of cyclic AMP. And we're going to see that that is a result of the PTS system activating the adenylyl cyclase enzyme. So when cyclic AMP it is available, that cyclic AMP can bind now to this cyclic AMP receptor protein, or CRP, which also in other books is called the catabolite activator protein cap. You're going to then produce cyclic AMP. Remember, adenylyl cyclase is the enzyme that produces cyclic AMP from ATP. And the PTS system is going to activate or inhibit that protein. When glucose is high, it inhibits the adenylyl cyclase, and therefore we have low amount of cyclic AMP. When glucose is low, the inhibition doesn't happen anymore, and the dilynyl cyclase is now high, and it can produce high levels of cyclic AMP. Another way why catabolite repression works is because the PTH system can also block the transport of glucose by blocking the glucose permease. So in here, the transport of glucose, it is tied together to the control of the expression of different operons.